Thank you everyone for coming this evening for this Sangwell Club talk. It's my great pleasure to invite and introduce Dr. Alex Kwong to the University of Cambridge. So Alex is a welcome senior research fellow at the University of Edinburgh, and he's been working on longitudinal models, particularly with youth internalizing problems with a focus on depression for many years now, close to a decade I'd imagine now. And his group is interested in psychiatric epidemiology, longitudinal data, and the integration of genomics into these methods. His particular focus of this grant is studying the causes and consequences of youth depression. And as a part of this, Alex has already sort of launched some very helpful, easy to use software that he promises to sort of give us a demo of, which is also sort of funded partly through the Wellcome Trust Data Prize Award. Thank you again for joining us today, Alex, and looking forward to your talk.
Thanks for the introduction. Thank you very much for having me here. Pleasure to be here and see all of you, and thank you very much for coming to this talk. So, as Varun very kindly introduced, this talk is about leveraging longitudinal population data to enhance understanding of depression across a life course, which is quite a lot of waffle, but I'm hoping by the end of it you'll understand what I mean. So, I'll just talk you through the three themes of what my group does up in Edinburgh. We're really interested in using longitudinal data to understand more about psychiatric epidemiology, which is about basically the understanding the causes and the sort of prevalence of mental health disorders within the population. I'm probably just going to focus on youth depression in this because we're working across quite a large sphere, anxiety, schizophrenia, but I think just to make it concise, we'll just focus on this. But if you have any questions about that kind of work, I'm happy to talk to you about it at the end. We're also really keen on leveraging longitudinal data in combination with genetic data, which is quite an emerging new field where we can learn more about what genomic influences and what environmental influences are impacting on certain traits and how that might affect across development. And then the final sort of thing that we're peripherally interested in is the way in which we can improve access to some of these longitudinal methods. They're really powerful, they're really exciting methods that can be leveraged to understand more about who's at risk of a particular disease, when they're at risk and why they're at risk, but what we want to do is make sure that we're allowing an equal playing field so that researchers who may not have the experience that other researchers have, we want to make sure that they're able to access these methods and use them as well for their data. So I'll start with a very broad background about depression. So mental disorders in general are a leading burden of disease worldwide, and this has been increasing in the last 20, 30 years. So we can see just down here, in 1990, depressive disorders were the 19th leading disability of disease worldwide. And then you also had things like self-harm and other things like schizophrenia, anxiety, making up the sort of top 200, 300 leading burdens of diseases. But depressive disorders are the highest in 1990, and that's actually risen to 13th in 2019. So the most recent estimates show that depressive disorders are one of these really key burdens of disease and the most impactful non-communicable disease on top of things like self-harm and anxiety. And the reason why this is important is because depression is a key mental health disorder across the life course. So if you look at all ages for disability adjusted life years, you will see that depressive disorders make up the most important mental health disorder. And this kind of tends to be more impactful from about the age of 15. So between the ages of 0 and 14, childhood and early adolescence, it's more conduct disorder, anxiety disorders that are making it up. But from the age of 15 onwards, depressive disorders are the leading burden of mental health, leading mental disorder across the life course. And we can say all of these things, but actually it really hits home when we start thinking about who this impacts and what this actually means in a day-to-day setting. So it's quite hard to get accurate prevalence estimates, but recent figures suggest it's about 1 in 3 women and 1 in 5 men go on to have depression, and that's about more than 280 million people worldwide suffering with depression. And that's just people who have a diagnosis. It's not people with sub-threshold symptoms, so people who don't quite meet the threshold, people who are queuing or lined up for services who haven't had any support yet. It's quite a significant burden of disease, and it's not just that having depression itself is bad, because it is, but it's also associated with a number of comorbid diseases, so anxiety, schizophrenia, cardiovascular diseases, but also it's associated with later problems in physical health and later health behaviours, so smoking, drinking, social functioning, and other social behaviours. So it's really important to try and capture and prevent depression early on, and to make sure that we can have good prevention and intervention schemes so we can stop this sort of cycle. And for many people, this happens when they're young. So depression is really, really prevalent during young people. And so we have good meta-epidemiological evidence now going through different studies and combining them all to show that this period of youth, which we could say childhood, adolescence, early adulthood, is a really key, important, critical area. And I think so what this figure shows here, that the peak age of symptoms, when symptoms first start appearing for young people, it's about the age of 15.5. The peak age of onset, the age where that onset is increasing most rapidly for people, is around the age of 20. But for people who've ever had, so 50% of people who've ever had a depression diagnosis will have had that onset before the age of 25, which is quite a startling statistic. It's just showing the key importance of this area. And if we think about when and how it's changing across this period of youth, we can see that in comparison to other diseases or disorders, ADHD, alcohol use, autism, conduct disorder, they're all kind of staying quite stable or they're getting lower throughout childhood and adolescence, whereas depression and anxiety as well are increasing throughout this period. So whilst other diseases are getting better, depression is increasing and rising. And this is quite devastating for later downstream outcomes. And as I said before, when you break it down into these periods after the age of 14, age 15, between 15 and 24, it's the second most biggest burden of disease for young people. And so what we can do is we can start to look and see what are the potential consequences to really kind of hammer home why this is important. And what I just want to draw you to is some cohort work here that's done by the Wolfson Centre in Cardiff. And what they showed is that if you group people into different trajectories of depression across childhood, adolescence and early adulthood, you see that these more severe groups, this adolescent adult increasing group, this green line here, and this blue line, which is the adult increasing group, and so adolescent persistent group and adult increasing group are more likely to have mental health difficulties later in life. They're more likely to have this greater functional impairment, and they're more likely to have suicidal self-harm ideology. And that corresponds to both of those two groups in comparison to groups that just have a bit of depression during adolescence and then get better, and those who don't have any symptoms at all. And probably what's worse about this is that there is consistent evidence now from across the globe that depression is getting worse. And this was happening before COVID-19, so we don't actually know how much of an impact COVID's going to have on these already increasing estimates. So why is it that depression is getting worse? Why is it that we have all of this knowledge and we have all of this understanding about depression, but yet it's still rising, it's still very prevalent, and we still haven't really made any progress in regards to treatment or interventions in the last 30, 40 years? The reason now, and more globally, not just necessarily for adolescent depression, is that there is now a greater understanding that depression is an incredibly complex disorder. It is comprised of both genetic and environmental influences. And previously in the past, when we'd say genetic or environmental things, we think about candidate genes. We think about specific, we used to think of depression as a single monogenic disorder. There was one gene for depression. And for years and years, even going back as far as 10, 15 years ago, we were focusing on serotonin transporter genes as a way to thinking if you have two alleles of the serotonin transporter gene and you suffer childhood trauma, you're going to have depression. It was never going to be that simple, and now we have consistent evidence that depression is a polygenic disorder. There are multiple genes, each having little tiny effects that, when aggregated, give a good representation of someone's genetic risk for depression. And that's indexed here by this Manhattan plot. Each one of these little green dots here going above this red line is a genetic variant, a single nucleotide polymorphism that we all have. Each one of these is a genetic variant that is associated with depression. And there you can see there are hundreds. In this case, there are 42, but I'll go on to show you some more about that in a minute. But also there's this great understanding that the environment is incredibly important. We see here that there's a list of 30, 40 that I've just picked out here of potential risk factors or protective factors that are associated with depression. When taking all of that together, there's this complex environmental picture and this complex genetic picture that interplay with one another, underpin one another to exert their influence on depression. So we need to have a better understanding of that. The other thing is that it's not just that having depression is a bad thing and then you go on to have depression once and that's it. Depression has very time-specific causes. So at any given point, there could be a number of things that could be underpinning depression. So I've taken this from a recent review that says that at the very basis of the understanding of what causes depression, it could be genetic liability. But as you progress throughout the life course, things start to be more important at given stages. So for instance, chronic physical illness, childhood anxiety, parental depression, these things could all be influencing themselves in early childhood. As you get older, you go through school. As you get older, you get through jobs and financial insecurity. Even older, you might have divorces and separations within the family, and then obviously spousal death as you progress further along down this line. So all of these things could be time-dependent impactors on depression. And this is going to have implications for how we develop treatments and preventions. On top of that, there is also this consensus that depression is not just the presence or absence of depressive symptoms. There's a specific, well, there are multiple number of ways in which depression can be exerted. So for instance, you could have single depression, you could have recurrent depression, you could have seasonal depression. On top of that, the onset could be early, it could be late, it could be during postpartum, it could be mild, it could be severe, it could be adolescent onset, it could be later adult onset. So all of this is another complex picture that adds on to the package. So how do we leverage all of that information, all of that understanding to try to understand and improve depression for people? So there are multiple ways, but I'm going to talk to you about the power of population data and the exciting methods and opportunities that are available for us to leverage some of this recent advances in data, recent advances in methods that can allow us to understand more about depression and specifically depression in young people. So population studies, they are very, very important for our understanding of health in general. But in terms of mental health, we have a specific way of doing it. For things like depression, there's no reliable or robust biomarkers. We're relying on people to tell us how they feel. And so what we can do to, obviously that has a number of limitations, but in this kind of case, you can ask someone a questionnaire and you can say to someone, at a given time in the last 2 weeks, how did you feel about something? Or we can extract a diagnosis record from health records or something like that. But what we can do is we can assess that multiple times across someone's life. And if we assess that multiple times across someone's life, we can actually begin to build a picture of what they're like, and then we can understand when something might be impacting on them at a given time point. And so just looking at some example here, so in a study like the Adolescent Brain Cognitive Development Study, so ABCD study in the US, they asked questions based upon the brief problem monitoring, so things like I felt worthless, I felt fearful, anxious. You can ask that multiple times, and they're really quick and easy ways to just quickly tap into someone's, in this case, depressive symptoms. At the same time, you might have something like the Children of the 90s study, or Ausbach, who ask a separate set of questions, and what they can be done is they can be asked across a specific period of time or across a life course, and we can use that information to understand more about someone's depression at a given point. But on top of that, we have the ability to then harmonize and replicate information from different sources. So for instance, you might have information on depressive symptoms in the Children of the 90s study, which I'll talk a lot about in this presentation. You might have it in different cohorts from the US, cohorts that are born 10, 20 years later from other cohorts, older cohorts which can still provide information, and then twin studies, and then other family studies. And the ability to harmonize between studies, the ability to replicate, so if you see something in one cohort and then you see it in the other, allows you to triangulate evidence, allows you to look at your question from a number of different viewpoints. And if you get the same answer, it's really quite exciting because it's more likely to be truer signal than if you just examined it in one cohort alone. And something that I'll try to focus on here is heterogeneity across these cohorts, differences in these cohorts, differences in measures, difference in the number of times people are assessed, differences in the instrument that is capturing depression. But if you still see signal through all of that in the persistence of that heterogeneity, then you can be more true that what you're observing is more likely to be a true effect. At the same time, you've got all this beautiful data, but you also have the advancements of methods. And we're in a really exciting time within genetic epidemiology now where we have access to these really large-scale data sets. So UK Biobank, you might have heard of, a data set of 500,000 people, each of them been genotyped, each of them with phenotypic information as well, information on health, social, economic data that exists. And what we can do is we can start to do large-scale genome-wide association studies. Again, what I talked about earlier, looking for common genetic variants that differ within the population that are associated with a given trait. And we have big consortium efforts now that are trying to pull together different large-scale data sets. So something like the Psychiatric Genetics Consortium is one of them. And I'm a member of the Major Depressive Disorder Working Group, where we're trying to pull together these big studies from around the world in different populations to try to understand the genetics of depression. And what that allows us to then do is to build this really rich resource of genetic data, which we can then apply at an individual or a local scale. So for instance, you can take rich genetic information, as shown in here, and create something called a polygenic risk score. A polygenic risk score being multiple genetic variants that can be indicated for one person. So aggregated and tallied, so that higher genetic risk is bad for a given trait and lower genetic risk is better, for instance. And altogether, these will bring all of this together, this exciting opportunity in methods development, this exciting new data that is available, will allow us to understand more about depression. And then the final thing is why longitudinal data? Why not just cross-sectional data? Why not just taking questionnaires and applying it throughout the population and just seeing how that goes? So you can do that, but my belief is that longitudinal data is a remarkably key tool for understanding about depression, because repeated assessments of the same period of time, repeated assessments of the same person over a given period of time will really give you an insight into the window of their lives. And also, if you have multiple assessments of depression in this instance, or multiple assessments of smoking, income, whatever kind of trait, if you take that in a longitudinal perspective, you're more likely to minimize measurement error and you're more likely to increase statistical power. And the reason of that is because 5 measurements from myself at 5 different times are more likely to be correlated than if I picked 5 different measurements from 5 different people around the room. And the idea is that you can leverage that correlation within individuals to improve power, improve signal. So what a typical study might look like is you might have these questionnaire items assessed at a given point, maybe at age 10, age 15, and age 18. These form a summary score of depressive symptoms, anxiety, psychological distress, and we get some descriptive information for a given person. Chuck that together with the rest of your population and you're able to estimate a subject-specific individual trajectory, but also a population average. And this can be really helpful for understanding how the population might change or what specific groups are at risk. So as I say, these longitudinal models allow us to identify who is at risk, when they're at risk, and then why they're at risk. And that's kind of what I'm going to talk to you about, some of the work that we've done on this so far. So the first one that I'm going to talk about was something that I did during my PhD. And what we did is we leveraged this repeated information from a study called Children for the 90s. It's a birth cohort located in the south of Bristol. It's 14,000 families that have been followed up since the mother gave birth in 1991, 1992. And they've been followed up for the last 30 odd years and have repeated assessments of depressive symptoms from about the ages of 10 to, in this case, the ages of 22. And it's 8 time points. And what that allows you to do is to estimate a trajectory for each person and then the overall population level trajectory. And what we were quite interested in here was just to see let's take a really key important risk factor for depression, sex differences. So we know that there's this preponderance of females being more likely to be susceptible to depression, but how does that manifest over time? And what we showed was that for around about the ages of 10 and 11, late childhood, early adolescence, males and females were roughly about the same. Maybe some argument that males were a little bit higher. But as time goes on, you see that this female trajectory gets much higher than the male trajectory. And we were quite keen to show that as some sort of monitoring of how males and females deviate. But what we also did is these trajectories allow you to go into a little bit more detail. So what we also estimated was the age of peak velocity, which doesn't really make any sense when you say it like that. But essentially what it is, it's trying to identify the period or the timing when depressive symptoms are getting worse at the fastest rate. So before they're getting to their maximum, which is around here for girls, about age 19 for girls, age 20 for boys, we can start to see where those symptoms are increasing at the fastest rate. So maybe that's an opportunity where we can implement some sort of intervention or prevention strategies to stop them actually getting to this local maximum. And what we found was that there was about a 3-year difference between this age of peak velocity for girls and for boys, which maybe seemed to correspond with pubertal timing. So we know that girls are more likely to go through puberty earlier than boys. There seems to be this correlation between those two things. That earlier pubertal timing in girls may be associated with this increased rise in depressive symptoms. At the same time, we can take that kind of model and also look at how genetic risk is associated with these different trajectories. So we have this risk factor of sex for males and females, but also what about looking at polygenic risk, looking at one's susceptibility for risk for depression, for anxiety, for schizophrenia, for neuroticism, and how does that manifest across the life course? And this was something that I did earlier in my postdoc, so we got some funding from the ESRC to sort of combine these genomic methods with these statistical methods. And the argument we made was that you can run this, if you have 9 time points of depressive symptoms, you can run this cross-sectionally 9 times, but it gets really messy and it doesn't give you all of this information that you could be actually, you could be maximizing all this information to learn more about your data. So we can see here that generally the pattern is, as people get older, there is a stronger association between polygenic risk for a given trait, so a given psychiatric trait and depressive symptoms. But that's as far as it will tell you. If you apply it to a longitudinal repeated assessment model, a longitudinal trajectory as it were, you can start to infer more about when genetic risk for someone might actually start influencing their trajectory. And we can see here that around about the ages of 14, 15, we start to see that people with a higher genetic risk for depression start to deviate from people with a lower risk for depressions and deviate from the people with just about average levels of genetic risk. And we see that those trajectories sort of, they sort of stay wide as people grow older. They don't necessarily come back in, they don't necessarily recover. So actually, we're sort of saying that this age of 13, 14 could be a really important area for the manifestation of genetic risk on how people go on to develop. And what we also wanted to show here is that traits like depression, anxiety, schizophrenia, neuroticism, they're genetically correlated, they're phenotypically correlated. People who have depression more likely to have neurotic symptoms, people with depression more likely to have schizophrenic symptoms. But just because they're genetically and phenotypically correlated does not necessarily mean they're going to manifest in the same way. So what we actually showed here is that just because they're genetically correlated and someone might have a higher genetic predisposition to schizophrenia does not necessarily mean they're going to manifest depression symptoms in the same way. So for instance, we don't see that having a higher genetic risk for schizophrenia is associated with a more severe trajectory. But neuroticism and anxiety do tend to have a higher trajectory. So those are the genetic risk and that's the environmental risk. But what about generational differences? I talked earlier that depression might be getting worse in young people as time is going on. So this is something that we did with Cardiff, and what we did is we leveraged 2 longitudinal data sets. So there's Children of the 90s study that I talked about, and then the Millennium Cohort study. And the Millennium Cohort study were born 10 years later, around about the year 2000, as the name suggests. And what we did in the study is we used the same measurements, the same sort of instrument to capture emotional symptoms, but they're just measured 10 years apart. So for instance, we have trajectories within Ausbach, which are the red line. We have trajectories in Millennium Cohort study, 10-year differences, but assessed at the same time for each cohort. So for instance, we have an SDQ time point that would have been assessed in about 1996 in Ausbach, and then again 10 years later in about 2006 for the Millennium Cohort study. So it's this time trend, it's this generational difference. And what we wanted to know is, are children who are born a bit later on, who are growing up with improved access to social media, improved access, maybe a generation of inequality in wealth, are they more likely to have greater depressive symptoms or greater emotional symptoms compared to the generation that were born before them? And what we showed is that this cohort that were born younger have a much higher emotional symptoms from about the age of 7.5. And this gap doesn't tend to shrink as time goes on. This younger generation are going to have these more persistent symptoms. And what was quite nice is we were able to sort of identify the scores at those given ages to work out where this difference was really coming in. And yeah, as from about age of 11, this difference was gradually increasing and remaining separate. But actually, what was more startling is that when you look at sex differences within those different groups, you see that it's these younger females who are at greater risk of emotional symptoms as time is going on. There may be some evidence that the older females are catching up, but we don't have that data collected at the point to work out if they were actually, that difference was shrinking or not. But what we can see is that these younger females have much higher trajectories than their older counterparts, and of course with the males as well. And we speculated a few things that might be responsible for this rise. Mainly the access to social media might be one of them, but not possible to test. and then these generational inequalities that might be widening as time is going on. I talked earlier about a complex gene-environment interplay, that the environment is heavily dependent on genetics, and there's this interplay and this interaction between the two. So what we did is we took a different method as to what I've described before. We now use growth mixture modelling, a data-driven approach to derive groups of trajectories that we expect to be present in the population. So for instance, we might expect a group of people, or the largest group of people within the population, to have low depressive symptoms. But then we might also observe groups of people that have higher depression trajectories that persist throughout childhood and adolescence, or those who start to emerge around late adolescence and early adulthood, those who start off quite high but get better, and those who just have this period during adolescence but then get better as time. And so what we did with my colleague Jose Lopez is we wanted to sort of make sure and validate these trajectories and see what might be the underpinnings of them. So in his paper in BJ Psych Open, Jose spent a lot of time trying to validate them and showed that these more severe trajectories were more likely to be correlated with depression diagnosis at age 15, age 18, and at age 21, and they were also more likely to self-harm compared to individuals in these lower trajectories, the blue line and the grey line. Also, we showed that individuals in these higher trajectories, this grey line and this orange line, were more likely to be not in education, employment and training. And some of the estimates that we pulled out, they were 7 times more likely to be not in education, employment or training compared to those lower group people. And then looking at the risk factors that are associated with these different trajectories, we took basically an a priori approach where we were looking at a bunch of plausible candidates, that being female, genetic risk for depression, postnatal depression, partner cruelty to the mother, childhood anxiety and bullying in early childhood. And what we found, which I think was quite a nice thing, is that these childhood and limited trajectories, this blue line down here, and this adolescent limited, this one that makes the sort of the arc, these were not associated with the genetic risk or mum's depression, or mum's postnatal depression. So maybe these are more environmentally driven trajectories compared to the early adolescent onset and the childhood persistent trajectories, this orange line and this dark persistent line here. These were associated with the polygenic risk score for depression, and they were associated with postnatal depression as well. So maybe there is this genetic or this familial risk that is passed on from generations that could be underpinning the more severe patterns of depression. And what we also found was that trajectories like the persistent trajectory and the early adult onset were also quite associated with things like bullying and child anxiety, and of course female was also associated with these higher trajectories as well. So there's this complex underpinning behind these trajectories that's quite hard to tease apart. And then this is some work that we've been doing, led by my PhD student Poppy Grimes, where we're trying to combine different sources of information to see if we can get a consistent pattern. So we're using now data from that Children of the 90s study that I talked about, but also data from an even younger cohort, the ABCD study, and that's in the US, and these are about 18 or 19 now. So it was about the mid-2000s when they were born. And so what we've tried to do is we've tried to replicate the trajectories in both that even younger cohort and then this older cohort here, the ASPAC study. And what we see is that using different measures, a different measure for capturing internalizing symptoms called the Brief Problem Monitoring Scale, and then we've got this short mood and feelings questionnaire on the right here, which is the ASPAC Depressive Symptoms Study. What we see is very similar trajectories, even when we're trying to take into account the fact that there's a different number of measurements, there are a different sample size, and there are a different cohort altogether. We see relatively consistent evidence that the trajectories are the same. And what we did is spend a lot of time trying to validate these trajectories because it could be an artifact of the statistical programming that we're doing. But what we seem to find is that they both have these more severe trajectories, this increasing and this persistent trajectory, are more likely to be female. They're more likely to suffer from sleep disturbances. They're more likely to have had a diagnosis of depression. They're more likely to have experienced bullying, and they're more likely to come from poorer socioeconomic status. So we're reasonably confident that these trajectories that we seem to be replicating across both cohorts are valid. But what we also wanted to do is take it a little bit further. So I talked before about genomic risk being quite important, and I showed you that individually depression risk, neuroticism risk, schizophrenia risk can be important for the development and the manifestation of these depression trajectories. But where there are new methods and exciting new paradigms that we can test, where we can take a spectrum of genetic risk. We can build a sort of a factor model where we load different genetic risk or different genetic variants onto an overall factor, or a series of different factors to test out our hypothesis about what could be underpinning these different trajectories. So here on the left we have a common factor model where we've just taken genome-wide association studies, summary statistics, these summary statistics of single nucleotide polymorphisms for a given trait, and we've loaded them onto a factor. So we're basically just summing them all up and putting them onto this overall spectrum of risk. In the second model, we considered a correlated factors model where we thought maybe depression, neuroticism and anxiety might load onto a mood factor, bipolar and schizophrenia might move on to a psychotic factor, and ADHD and autism spectrum disorder might load onto a neurodevelopment factor. And the final one was just having an extra level on top of that. We call it a hierarchical model. So you've got these factors that load onto this overall arching p-value or p-factor of risk. And what we found is that if you think about these trajectories that were the same for both Ausbach and ABCD, this persistent trajectory was associated with all of those individual polygenic risk scores, but over and above that, it was associated with these hierarchical, common, mood, psychotic, and neurodevelopment parcels. So what we're seeing is that the aggregate score of all of these different genetic risks for these different traits seems to be a really robust marker for genetic risk affecting these different, more perverse trajectories. And we see this in both this ABCD cohort and in the Ausbach cohort. And what we also see is that throughout the rest of the different trajectories, the increasing trajectory, for instance, and the decreasing trajectories, the evidence is less consistent for these individual polygenic risk scores, neuroticism, anxiety, depression, but it's very consistent for this hierarchical and common model, and we see this consistently across these different studies. I would say that I urge a little bit of caution for this particular increasing one because we're only going up to age 14 or age 16 in Ausbach, so we haven't actually got into those later periods of maturation yet. But what we do see is certainly in these early life patterns, so a persistent pattern of depression across adolescence is associated with this higher genetic risk. And I should say that most of this study to do with genetic work has been based upon European ancestries. So for instance, we get a lot of our data from 23andMe, which are heavily based upon European ancestries. But there is now this big drive to move towards more diverse samples, understanding that depression doesn't just affect European people, it affects people of all ethnicities. But unfortunately, that data just isn't there at the moment, but we're starting to get better in terms of genotyping and collecting phenotypic data for more diverse ancestries. And so what we can start to do in this manuscript at the moment is look to see how genetic risk, not just in Europeans, but in Africans and people with admixed ancestries and in Southeast Asians, how are they associated with these trajectories? And I'm not going to go as far as to say they're not associated with these trajectories, but there is good evidence that probably with an increase of power, with an increase of sample size from our original GWASs that are not just based upon Europeans, if we can increase those GWASs for people of African ancestry, for admixed ancestries, we'll start to see genomic risk being associated with these more perverse trajectories as well. So coming into this bit about new data and new opportunities that I talked about earlier. So these polygenic risk scores, which I talked about before, were made, so especially the depression one, were made on sample sizes of maybe about 1 million people. But we're moving into this really exciting area of genetic epidemiology now, where people want to give their, well, firstly, people want to give their data. There is this exciting opportunity that people are providing data and we're getting it at its source, but also there is initiatives like the Psychiatric Genetics Consortium, other consortiums exist around the world, which are pooling together data sets. So for instance, this just gives an example of the green line gives an example of where depression GWAS, genome-wide association studies, have been over the last 15 years. So we start in 2012, there was the first major depressive disorder GWAS, which unsurprisingly did not find any genome-wide hits, given that the sample size was about 10 to 20,000. Fast forward to 2018, Naomi Ray put out a GWAS of major depressive disorder where we started to identify a few genome-wide hits. This was followed later by David Howard, who found 102 genetic variants. And as we're going along, we're slowly getting more and more cases, and then that takes us up to about here, which is where the recent GWAS is coming. But importantly, we're starting to add more and more ancestrally diverse samples. So for instance, we're starting to add African, East Asian, Latin American, South Asian people into our GWASs, so we can start to get accurate genetic risk for these populations as well. It still pales in comparison to the European ancestry, but we're trying to make this a more accessible and a more diverse group of people to include within our GWASs. This consortium effort of bringing people together, bringing these studies together, is paying dividends. So in the previous GWAS, one of the previous GWASs I've been using, we found 102 genome-wide significant hits. These are genetic variants that are robustly associated with depression. 102 was very good at the time. We had good heritability, SNP heritability. We were quite excited about that. But obviously, we were very naive because fast forward from 2019, fast forward 5 years, and we're just in the process now of submitting this work led by my colleague up in Edinburgh, Mark Adams. And we now have 629 genome-wide significant hits, okay, an extra 500 more. This leads us to understand more about the heritability of depression, but it also allows us to do a lot of downstream things. It allows us to understand more about drug targets, about the biology, about the underlying genes, things that we can then use for more practical purposes, not just necessarily for observational or descriptive epidemiology. But where it becomes really exciting for me is that we were able to then use designs that are quite powerful within traditional genetic epidemiology or epidemiology, and we can use these methods and apply it to youth depression. So for instance, we can use a method called Mendelian randomization, which we couldn't used to before because we just didn't have enough power in our genetic variants. So what we need, I won't go into too much information about Mendelian randomization, but essentially it's a form of instrumental variable analysis, which is very popular with people in economy. But essentially what we're saying is we need to have an instrumental variable which is associated with an exposure. So in this case, we want to have genetic variants that act as a proxy for our exposure variable, which in this case is depression, and that exposure then has to be associated with our outcome. And there are a few assumptions that we need to make here. So firstly, that Z, which is our instrument, is associated with depression. Previously, this didn't used to exist. We just didn't have the power to do that robustly, but now we do have that power. We want to make sure that the association between Z and X is independent of controlled confounders, and we can test for that now because we have some of this greater power. But then also we want to basically assume that Z is affecting Y only through X, so it's kind of like a mediation bit in that bit. We want to make sure that any relationship between our instruments and our outcome is associated through depression. And so what I did in this study is I just used this information. I used this new method and these new data to look at the causal role of adolescent depression on later outcomes. And what I did is I used the triangulation approach. So what I did is I looked at just very simple logistic regression, just looking at the odds of things like financial problems, poorer health, daily smoking, physical activity, high TV use, lower income, alcohol use, and hospital spells. I looked at those outcomes of adolescent depression, the experience of having adolescent depression, what does that result in later on? And I did this just using very simple multivariable regression in an observational approach. But then I used this Mendelian randomization framework, a one-sample MR framework, to try to support that, because any one of these methods by itself could be potentially biased, but then trying to support it with a new causal method showed sort of some evidence that depression may be causal adolescent depression may be causal for financial problems, poorer health, smoking, and any low physical activity. You can also use larger scale data. So I also used data from UK Biobank, not an adolescent cohort by any means, but it has that data, it has genetic data, it has this phenotypic data. And what I did is I used some two-sample Mendelian randomization. So I'm not using any individual cohort population data, I'm just using genetic data to look at a causal effect of depression onto some of these risk factors that I pulled out, some of these outcomes that I pulled out in the ASPAC study, in the Children of 90 study. And I see consistent evidence that depression genetics are causally related to things like financial problems, poorer health, and maybe cigarettes per day. So what we're doing is we're looking at it from the lens of three different ways. We're looking at it from observational epidemiology, classic regression. We're looking at it from one sample MR, which supports the the observational epidemiology. And then we're looking at it from 2 sample MR as well, just for providing a further lens on that. So that was quite exciting, and we're hoping to just try and follow some of that up with some other cohorts now, just to provide an extra bit of evidence. And then what my group are really interested in, as I said at the beginning, are the process of longitudinal genomics. How can we integrate longitudinal data with genomics to improve sort of inferences? And one thing that I've been thinking about for a long time is trying to improve the power and the precision of GWASs by making sure that what we're measuring, the phenotype that we're measuring, is actually robust. And so if you take a questionnaire and assess it at one point at a time, you're going to be subject to some measurement error. If you ask how someone's been feeling over the past 2 weeks, if they've had a really bad day yesterday, they're going to score quite highly today. If they had a really good day yesterday but a really rubbish few days before that, they might be subject to some noise and some measurement error. But if you take multiple assessments, you're more likely to find the balance or the true score of someone's individual than if you just took it at one point alone. So what I've done is I've tried to highlight this through a series of different assumptions. So just taking this baseline model, this one where you just say, I'll take the first time point in which they completed it, and I don't care about the rest of the time points afterwards. I don't care about anything that happened before. If I miss the baseline, I'll take the next one. That's the first time point model. You could also then just say the mean of all of those occasions. So for instance, in UK Biobank, there are up to 7 occasions where the same thing has been measured repeatedly. The PHQ-2, a measure of psychological distress, depressive symptoms. So you could take the mean of each of those occasions. The other model we've been quite interested in is a shrinkage model, a precision-weighted shrinkage model. You run sort of a multi-level model without looking at time, or you don't really care about how time is changing. You just want to shrink everyone closer to the mean so that people at the extremes are more adequately captured. And then the final model was sort of that p-factor model that common factor model that I spoke about earlier, where we've got all of these, we've GWAS each occasion, get an idea of the summary statistics, get the genetic variants from each occasion, and then we load them onto this overall p-factor, hoping that we can try and capture some of the overlap to increase some of the power. And then what we've found is that in the baseline model, we find that the sample size is the same for everyone, so that doesn't change. But what we find is that in the simple baseline model, we find 28 leads single nucleotide polymorphisms. We find 62 independent SNPs, and we find 142 genes, and we've got a heritability of about 4.5%, which is pretty good. But what we do find, though, is that this mean model and this shrinkage method, these longitudinal phenotype methods, leveraging this repeated nature of it, massively outperform the simple baseline model. So if we just go straight to the shrinkage model, we have number of lead SNPs, 28 in the baseline. We have 62 in the shrinkage model. Independent genome-wide significant SNPs, we have 62 in the baseline. We have 169 in the independent significant SNPs. We have 142 mapped genes in the baseline model, 192 in the shrinkage model. And that's quite important because if we can map more genes, we can understand more about the biology. We can infer more about drug targets. And then the heritability increases by about 2%. If you plot them both on top of each other, what you can do is you can actually get an understanding of where these increases are coming from. We see this peak here for the shrinkage method that doesn't even get picked up in the baseline model. We see these much higher peaks in our genome-wide association studies that just don't get picked up in the baseline model. Yeah, so it's 121% increase in the number of lead SNPs identified, 173% increase in the number of independent significant SNPs. And just that improving that phenotype precision could go a long way into improving some of the power for GWASs that I spoke about earlier. Okay, so just coming on to the last bit of the talk now. So I said in the beginning that my group are really interested in developing methods and facilitating access to methods to researchers or people with longitudinal data that might not necessarily know how to run these models or don't necessarily have the time. And we aimed this project, so this was a Wellcome Data Prize that we were lucky enough to get funded for several rounds, where we pitched this idea that a clinician or a school teacher or a very early career researcher who has longitudinal data or data on pupils or data on patients might have this information that could be really important, but don't really necessarily have the skills or time to analyze it. And so you might have a data set that looks something like this, where each row is an individual person, then you might have 4 columns of a given mental health disorder, and then some age variables, and then some other demographics. how can you quickly turn this file into something that's interpretable and readable and understandable for an audience? You can do this through this tool that we've made. And hopefully it works. Whilst it's loading, there's a GitHub which you can download, and there's an R package if you're interested in that. But essentially what it does is you just take a data set. So we've got a simulated data set on here already, and this tool will automatically search through your data set to find the column which it thinks is your ID for your participant. And let's say you wanted to take 4 time points of a particular trait. So it'll look through your data set and try and find something called age or time or occasion. And maybe you're interested in modeling something called the Strength and Difficulties Questionnaire, which is a model of internalizing and externalizing symptoms. So what it will do is it will do all of the complex stuff that you need to do to run this analysis, but it'll run it in the background. It will do things like turn your data from a wide format into a long format so it can run multi-level growth curve modeling, and it'll tell you what it's doing as it's doing it through code. And what it will do is, if I just run the model, it will go very quickly from that Excel spreadsheet that I showed you into something that's a little bit more interesting. It'll plot the number of people at each occasion, it'll plot the mean, the standard deviation, the median, the interquartile range. It'll also give you this nice little plot so that you can understand the descriptives of your data. That might be enough to show someone else that you're a co-I or a collaborator that you're working with that this is what your data are looking like. At the same time, it gives you the model results. It'll tell you what model it's run in the background so that you can actually take that and say, actually, this is all too basic for me. I don't care about all of this. I want to know how to get into it, but I'm going to do it my own way. I don't want to run it through your tool. And it will also give you, so it will give you the R code so that you can replicate the analysis, and it will also give you the results from the model. And it'll give you some interpretation that we've worked with researchers, we've worked with clinicians, we've worked with school teachers to make sure that interpretation is quite accessible and doesn't allow you to overestimate or overinterpret your results. Maybe perhaps the most important thing is the ability to plot a trajectory through your data. And if I go into an interaction variable, so that's just plotting the mean levels of data, but maybe you want to know how that varies by a particular group or subgroup. So what you can do is say, I want it to run between, I want to see how results differ between males and females. And so again, it will give you the results, it will give you the interpretation of what those results mean, and then it will also plot a nice figure for you with confidence intervals so you can see if there's a difference between your two populations. And then what it also does is, let's say the output from your model is quite messy. Once you start going, so this is a very simple linear model, just for every 1 unit increase in age, the SDQ score is going up by, I don't know, 0.1, 0.2. If you go really complicated, let's say you go into really non-linear models, quadratics, cubics, where you've got people going up, people going down, the interpretation can be quite messy. So what we've done to accommodate that is given the option to be able to calculate what the score is for a given group of people at a given time. So for instance, you might be interested in estimating scores every 3 years. And so what it does is it calculates those scores for you and tells you what the difference is between each group. And then we've also been trying to develop other features which might help for interpretation. So we've called this area under the curves, the proportion of time a group of people spend with a given disorder. And then the final thing is that there's the ability to look at individual level trajectories in comparison to the mean. So for instance, you might have a group of people that you're specifically interested in. So maybe I'm really interested in 5885, this blue line here, is massively diverging from the overall population. So actually I can look and see how 5885 differs. Okay, it wasn't 5885, but the point is you'd be able to identify individuals who may be diverging from the overall population trajectory. So that's one method that we've been developing, and it's free to use as a Shiny app. We also have an R package if you can't upload sensitive data, and we've also developed a Windows and a Mac version for it as well. And yeah, one thing that's been really cool working on this project is we held a little hackathon with researchers from different universities who have access to their data sets. And what we did in an afternoon, in what traditionally would take me a week to maybe do this kind of analysis, is we asked people to bring some symptom data and some data on time and data on different demographics. And over an afternoon, we just told people how the tool worked, sent them away, came back in a few hours, and people had their own individual plots. And what this is really helpful is providing a framework from which people can then use and harmonize and try to replicate data. And we said, can you just look at see how trajectories might differ between males and females in your cohort data? And this is what people came back with. They came back with consistent evidence that around about age 13, age 14, we start to see these differences in mental health, in depression, emotional traits between different cohorts. And this is consistent across all these different studies that we show. But I think what's nice is that it was done very quickly. This might take a long time to do it, but the fact that we've provided this framework made it quite quick and accessible. So that's all I'm going to talk to you about. And yeah, I'll just leave you with a few things saying that hopefully I've shown you that youth depression is a very dynamic and complex problem, which has a very complex underpinning of genetic and environmental influences that affect, these genetic and environmental influences affect not only how depression onsets, but on the manifestation of time and how that persists across development. We've got these really new and exciting methods that are coming out and that we can leverage to really gain greater insight into what is going on, not just for youth depression, but for depression more generally. And hopefully this might facilitate a more precision medicine approach. Precision medicine is something that's very topical right now. In psychiatry, it's really hard to actually leverage that properly, but hopefully in the future, some of this kind of more biology-focused work will be able to infer some of these greater precision medicine approaches. And then just leave you with the concept that we need to have more diverse sample sizes, more diverse samples, more ethnic samples for different at-risk populations to really fully understand what is going on in depression. And I'll just say thank you to my team and then my list of collaborators, and I look forward to any questions you might have.
Thank you, Alex, for this really nice, comprehensive overview, both of genetic and glomming in longitudinal aspects as well. Also, a really nice tool. I'm sure it'll be super useful for people. So, let's see if anyone has any questions for Alex. Claire.
So first of all, thank you for a really, really interesting talk, really inspiring. In terms of methodological innovation, there are changes that are going on as well in terms of picking up on environmental experiences. So I'm thinking about experience sampling in particular. So not just having multiple time points over a period of years, but multiple time points over a period of days or weeks, so that then you can actually get some of the lability,
yes,
as well as just having more data
yeah.
per person. I'm also aware, I don't know if there's anybody here from psychiatry, but the Child Psychiatry Department have developed an adaptive testing of mood in adolescence, which could work really well with that experience sampling, because you won't get the same boredom threshold because you're asking them exactly the same question each day, because which question you ask depends on how they answered before. And I'm just wondering how you see those sorts of changes in measurement influencing or interfacing, if you like, with all the innovation that's happening kind of from the genetic angle.
Yeah, great question. Thank you. And that's one of the main focuses in my fellowship. So what I'm doing is I'm going into the experience sampling framework, and what we're going to do is we're going to embed those kind of experience samples within those different trajectories. So for instance, you will have people with the high risk, people with the low risk, people who've got better over time, and we're going to go in and ask them what's 2 weeks like in your life, and then collect more data, more questionnaire data, and then go back in again and just to try and do these sort of repeated bursts with, so you've got the long term scope, and you've got the short term one, and just seeing how does that affect, and then are there specific things that are going on in the day-to-day environment that might tell us more about why they're on these persistent trajectories, or why they're on these different courses. But yeah, I think that's the future. But there is a lot of people working in that sphere. I don't know how long that sphere will last.
University of Edinburgh, I assume you know Aisha.
Yes. So she's a collaborator on my grant. So she's doing this. She's the one who inspired me to do this repeated burst design. So yeah.
I was interested in your archetypal trajectories. It seems that you wait until people are of a certain age and you take some sort of mean depression score, and you then do some sort of a classification analysis, and you then take those classifications as a categorical independent variable, i.e. people who have this type or that type,
yes,
or whatever, and you then do retrospective analyses in which the statistical dependent variable can be anything. It could be actually something that was prior, it could be in the genetics, but there are other combinations. Is that the best way to do things? I mean, you've got 4 categories there. Is the matter perhaps not a bit more refined than that? How did you, what sort of cluster analysis did you do to get them? And would it be better perhaps to think of subclusters or express it in a totally otherwise dimensional space in terms of continuous variables that describe trajectories, which it's relatively elementary school mathematics to do that.
Yes.
It was unclear sort of what that had achieved, or how large clarification, how big effect sizes you were getting from pursuing that archetypal trajectory approach.
Yeah, of course. No, that's a great question. Thank you very much. So this, let's go with this one. This is the kind of model you're thinking about. Yeah. So they don't necessarily need to be. Well, okay. I'll start with there are 2 schools of thought within this kind of field. Is do we think that depression should exist on a spectrum, or do we think that within that does that spectrum capture enough? Is that spectrum too broad and then doesn't capture enough of the subgroups? So I'd like to think that the 2 methods that I presented, this one as well, this multi-level growth curve one, which is the spectrum essentially, and then this categorical group, I'd like to think they're quite complementary to each other. Yeah. So what we do here is they don't necessarily need to be independent variables. They can be dependent variables as well. They can be outcomes of the model. But what we do is essentially it's a data-driven approach. We feed the model into a data, and we start with k-means of cluster of 1. And we say, we have an overall, this is the overall population line, everyone was within that. And then we add basically groups to that, so we can say, is the model fit better for 1 group versus 2 groups, and then 3 groups versus 2 groups, 4 groups versus 3 groups. At some point, you get to a bit where there's no improvement in model, and you start to see weird things happening with the data. So firstly, the most common one is when the data doesn't converge, and then you think, okay, that's a classic statistical problem. Okay, maybe it's fine. So you keep going, and then you start to see maybe people where there's groups where there's maybe 2 or 3% of your population, and so maybe in a smaller study you might see 6 or 7 people in a group, and you might think that's maybe not robust enough. The key thing, which I think is quite good, is that these groups we tend to see, and why we tend to go with them, is that we tend to see these groups as being more common in clinical practice. So we do see a group of people who onset during early adolescence and then stay quite high. So a lot of it is based upon the clinical validation of those trajectories. So if we can be more confident that there is an early adolescent group, there is a later early adult group, there's a group that are consistently low, then we can be more confident those are meaningful groups. But at the same time, there are a lot of people who don't like this method because they don't think it captures that spectrum of risk. And as you could say, there are probably subgroups within each group. So where do you draw the line? It's a bit of a dark art, actually. This one.
Okay, thanks so much.
No, thank you. Great question.
Thank you. It's really fascinating. I was particularly interested in the transdiagnostic part, where if I understood right, you had a factor model for constructing PRSs, using multiple different disorders, and then this was more predictive of, I think it was the specific different trajectories on the
Yes.
the illustration of the PRS when constructed in the transdiagnostic or multi-diagnostic way. I just want to hear more about your thoughts. This seems important, and maybe does this say something about how we should be thinking about GWAS in general as a diagnostic?
Yeah, so I think what's quite nice about it is it just shows that there's a spectrum of genetic risk. So maybe we don't capture enough of it at the individual polygenic risk score level, but as a spectrum we can get more information about it. And then we can potentially use that in machine learning models, for instance. So if we can use a more powerful approach for capturing this genetic risk that might be more useful in personalised medicine approaches.
So not necessarily multi-diagnostic. Is it the multi-diagnostic part that's important, or the score, the fact that it's using?
I think I think you can probably do both. I think you can probably say that they're both important. I think what we were thinking here is that long term, you could probably incorporate. It'd be much more justifiable to incorporate a transdiagnostic, a multifactorial trait polygenic risk score into a prediction model, because then it's captured all of that genetic risk to begin with, and then you're not having to penalise for adjusting, for adding in more individual polygenic risk scores. Thank you.
When you were discussing the basic GWAS for depression, you suggested that it was polygenic in the sense that it was the cumulative effect of that each gene was having a small effect. Can you rule out the possibility, which is very plausible for, say, schizophrenia, certainly is probably the case for intelligence, that there are major genes, but they're different in different families?
Yeah, I think that's probably fair. I think, but do we? Yeah. So what we've done in this sphere already is just do very common genetic variants, ones that are very easily picked up when we're tagging people. I think we're now moving into the whole genome sequencing area, where I think we'll have that. So they've just released that data on the UK Biobank now. So I think there'll be greater power to detect more important genes in that. But I think for depression, we see probably a few maybe more important genes than others. But I wouldn't like to guess too much about if it's more important within families. That's not my sphere, unfortunately.
Any other questions? Good. In that case, thank you again.
Great,
Alex.
thank you.